Great. So, Keith, just to get started, I'm really excited to be here today with you. For those of you who don't know Keith, he's the CEO of the CureGRIN Foundation and also the host of several podcasts. One is called Unlocking Bryson's Brain. I think you have another called My Instruction Manual. So hopefully I this is my first and only podcast. Hopefully I learned something about podcasting from you. I'm just going to say I really recommend everyone listen to your podcast if they haven't yet. It's called Unlocking Bryson's Brain. And I was wondering, because probably many people haven't, if you could maybe just take us back to those first few years of Bryson's life, some of the challenges you faced around getting a diagnosis and some of the color around your decision to dedicate much of your professional career. I think you were a journalist previously, and actually really kind of switched gears into searching for a cure.
Yeah. So I guess it was almost 14 years ago now that my wife gave birth to our second son, Bryson. And, at first everything seemed normal with his birth and his growing up. His initial scores were good. He seemed like a healthy baby. But then when he was a couple of months old, we started noticing that he wasn't developing in the same way that his older brother had. So he wasn't holding his head up as strongly. He wasn't tracking objects with his eyes. We went to do baby pictures, and the photographer was trying to kind of get him to prop up his head on his fists, and his head kind of kept toppling over. So it was clear that something wasn't quite right in his early development. And at about 3 months, at his 3-month appointment, we told doctors about this, and they said, yeah, something is not developing right with Bryson. And they didn't know what. And that sort of started a nearly decade-long journey to find a diagnosis for Bryson. So during that time, he had MRIs and probably 15 different, actually it's probably even more than that. I would say at least 20 different blood tests to rule out different kinds of diseases. He had a muscle biopsy, he had EEGs. So, it felt like we were in the hospital kind of nonstop doing all of these tests. And we just got to this point where it seemed like it was probably some kind of rare genetic condition, but maybe we would never know what it was that Bryson had. And then we came across a doctor who recommended that Bryson get a whole exome sequencing test. And at the time, those tests, and actually they're still not paid for in the province of Ontario. We have a public healthcare system here, but that doesn't mean that everything gets paid for. And so we needed to get special permission for that test, and this geneticist was able to get it. And the tests came back and confirmed that yes, Bryson had a de novo variant, meaning he didn't inherit it from Laura or myself, and it was in the GRIN1 gene. And so, immediately on the way home, we started Googling to try to find out what that was. And there wasn't a lot to learn, but we found that there was a Facebook group with about 7 other members. We joined, and in the years since then, over the last 4 years, we've met many families around the world with variants in the GRIN1 gene and the 6 other genes that code the NMDA receptor. And about 2 years ago, we hosted a conference here in Toronto. Families came in from across North America, and we invited speakers from Toronto, at the University of Toronto and at the Hospital for Sick Children. And what we heard from them was this message of hope. And they talked about how they really believed that this variant, these genes could potentially see a cure, that the NMDA receptor it's well known to be responsive to medical therapies. And, we were in this fortunate position because unlike so many genetic conditions, this involves a single gene, not a bunch of different genes. And so, gene therapies could be possible, gene editing someday could be possible. And from that conference, I kind of decided to make it my mission to learn as much as I could about GRIN and NMDA receptors, and to kind of do what I could to bring researchers together and to connect families with researchers and clinicians and basically raise awareness. And as part of that, I got together with some other parents and started up the CureGRIN Foundation.
When did you all start the CureGRIN Foundation? Was that last year, 2 years ago? Was it before the before the conference? Because you started the conference, right? Or at least you were part of starting the conference that you mentioned.
I was part of, the conference in Toronto was before CureGRIN Foundation.
Right.
Yeah.
And what made you decide that you needed to start? Because you had this group of people organized on Facebook. Did you have any idea of what proportion of the total people worldwide, how many people actually have GRIN, how many families are affected by GRIN disorders? Was it, most people were in the Facebook group, or do you have an idea that there's actually, many more people out there?
Yeah. So, I think it's one of those things that can happen a lot with these genetic conditions that have only sort of recently starting to be diagnosed, where you start with just a handful and you think that it's an ultra rare disease. And, the more things go on, we find more and more people around the world. And, there's recently a paper that estimated that the number could be in the hundreds of thousands or even over 1 million that have pathogenic variants in one of these genes. So, which would still make it a rare disease, but not an ultra rare disease. And one of the things that I've learned is that, that's not necessarily a bad thing. A lot of rare disease people or parents kind of like to, wear that badge of honor, like the rarer my kid's disease is, the better. But in terms of getting biotech and researchers interested, obviously, there's a lot of interest now in rare disease, but there still has to be a critical mass. They want to know that if they're investing time and money, that there's going to be a potential return.
Yeah. How did you navigate getting that diagnosis in the early days from an emotional level? I think you said it was dozens of tests over probably a decade. And I remember from the podcast, one of the first tests that you had sent away to check for spinal muscular atrophy, they actually didn't send it, and you had a 4-month delay. And I imagine that must be excruciating, but it actually happens to I would say probably the majority of rare disease parents experience an odyssey like the one that you experienced. How did you actually handle that emotionally?
Well, I think, to be honest, my wife and I handled it in different ways. I was working full time, and I think that maybe I was able to sort of put it out of my mind a little more, where my wife was decided not to go back to work yet, and so she was at home with the 2 little kids and kind of, going to all of Bryson's doctor's appointments. And so I think it was, extra tough for her, but it's tough. I mean, it's, you feel like, and especially when, there came to a point for us where it felt like doctors, they kind of felt like they'd done everything that they could and were sort of ready to give up. And so we were really grateful that we found this doctor. And in fact, there's an interesting story that I tell on the podcast, that my wife found this doctor that seemed like the perfect guy at Johns Hopkins University in Baltimore, or Johns Hopkins Hospital. We were planning to travel there to meet with him. And then he said, hold on, I'm actually moving to Toronto. I just got hired there. So we were one of the first patients. Bryson was one of the first patients to go in and see him. So we were grateful for that. But certainly that search for a diagnosis can take an emotional toll.
What did he do that was so different from all the previous doctors or clinical geneticists? Obviously, besides having the wherewithal to know about exome sequencing and pursue it, what was it that made his approach so different from the others?
He was, at the very first appointment, we called him Dr. Cohn, and he said, no, call me Ronnie. He just really seemed to have a very patient-centric approach. And I think we'd heard from other doctors that maybe we would never get an answer. He told us, no, I think maybe, we will figure this out. And we'd had some very bad experiences with doctors. I talk about some of that in the podcast as well. But just, he listened, he had compassion. After we had this initial meeting where we got the first diagnosis, and it was just before the holidays. And he said, I'm actually going to be in the office over the holidays. If you have any more questions, come back and see me. You don't need to bring in Bryson. I've heard from other families who get their genetics report and like don't get to talk to a doctor or even a genetics counselor. So
Right.
just having that, we were really grateful that he kind of talked us all through it.
How did he actually help you navigate the healthcare system? Because probably at the time that test was thousands of Canadian dollars. I mean, it still is in a lot of places.
Mm-hmm.
Was there some kind of special program here in the UK? They're starting to make this testing free and a lot more widely available, but I still think it's going to take some time. How did you manage that?
Yeah, so I guess there's a process where we could actually apply, and he kind of facilitated all of that for us. He actually told us that if he couldn't get funding this way, then maybe through his research budget we could get results that way. But luckily, the province came through. But again, it's still not something that's covered. So the number of patients in Canada that have been diagnosed with GRIN disorder, we're about 1/10 of the US population, but we certainly don't have 1/10 of the diagnoses. So it's an interesting area where kind of like the private insurance in the US is ahead of what the government's doing here. And I think that, and so, if you have private insurance, that's a good thing. If you don't, it's not so great, obviously, in the US. But I think this is an area where the cost benefit really seems like it should be in the favor of paying for these tests, because if you just add up the costs of like all these one-off tests that Bryson had, it's got to cost more than the thousands of dollars that we was eventually spent for the whole exome sequencing. And now we're seeing that some of the GRIN genes are starting to be added to some of the panels for intellectual disability and for epilepsy. So it's possible that those weren't on any of the panels when Bryson got his diagnosis or when he did those panels, but it's possible now that families could get diagnoses sooner. And certainly we're seeing some kids diagnosed as early as 2 or 3 weeks now, which is new.
Yes. And I think what the calculations often fail to capture, in part because I think it's really hard to capture, is the non-medical benefit as well. I remember in the podcast you were saying your wife in particular was really needed an answer around whether it was a degenerative condition or not, right? So you knew how much time Bryson had, and I think that question, the answer to that question alone is worth its weight in gold, right? Even if there's nothing you could do from a health perspective, even that and it's very hard to quantify though, isn't it, from a healthcare system perspective?
Yeah. And even, Ronnie Cohn, the doctor the geneticist that I talked about, he sort of said that he had really come around where early in his career, he'd been the guy saying, let's not focus so much on the diagnosis. Like, what purpose is a diagnosis if we don't have treatment? And let's focus more on the condition and the person. But he said that, over his years working as a geneticist, he really came to understand that providing that diagnosis, even when there's nothing you can do, really can have such a huge benefit to the family.
Yeah, and I think one of the other, I did my PhD work in rare disease, whole genome and exome sequencing, and one of the things that I thought the group that I was a part of did really well, it's here in the UK called Deciphering Developmental Disorders, but they would have I think it was a yearly meeting that was open to participants and family members. And it was amazing to be able to meet people that would say, even if there was not a treatment available, to say that they found a group on Facebook like you did, for example, and they could speak to someone who had a child who was, maybe 3 or 4 years older than theirs, to understand, to have that peer connection or learning to understand whether what they're experiencing is different or the same as others. And I think the other thing that you all have done really well is it can be an amazing catalyst for research, because if you have 7 families in a Facebook group, there's only so much you can do. But if you have 100, 200, I think you guys have 1,000 people following you on Facebook, and you probably have a really active community now, that you start to get more people who are good at different things, right? You have an incredible gift of understanding the spoken word and journalism and those sorts of things. And you probably have other people in the group that bring different expertise. So you can start to build a coalition. Has that been your experience with CureGRIN?
Yeah, absolutely. And not only that, but also just having like the global reach. So, there's people who are living in countries where they've been able to talk to local researchers and get them interested in GRIN that we would never have thought of. So there's like a lot of research being done in Barcelona. And so there were researchers doing that on their own, but then there's a huge now group of patients that have come together in Spain, and because I guess they do genetic testing well there, and so there's a big group, and they work closely with those researchers, and that just helps drive it forward and helps those researchers build the case for funding and that kind of thing.
There's a question that's come through on the chat that I think would be really good to ask right now. Jesse says, what do you think about trying to get the federal government to allocate funding to Canadian universities who are willing to do these genetic tests for free? Not only would it be cheaper than alternative methods, as many universities have the infrastructure, but it would further Canada's research through our own institutions.
Yeah, I mean, I think there's probably a lot to think through in that. I mean, I think probably if it's something that's happening at universities, there need to be a process where patients or patient families are consenting to share their data or make their data available for research in a way that wouldn't necessarily be if it's paid for out of healthcare. So I think there's probably pros and cons. I think the best case probably is that patient families can still maintain their own privacy, and it's actually paid for out of a healthcare budget.
How on that privacy and data point with your work at CureGRIN, what stage are you all at? Do you have biotechnology companies that are now actively seeking your partnership out because they're developing potential treatments, or are you needing to still build those relationships? Have you started as a group collecting information, building natural histories to aid this sort of thing, or where are you at in that process?
Yeah, I would say somewhere in between. So we've, for the most part, we're still at the point where we are reaching out to biotech companies. There's certainly some that have therapies that target the NMDA receptor. And so they're very, very interested in hearing from us. Some of these companies kind of build compounds, knowing that they do important things in the brain, but not really being sure yet where they're going to apply them. So, a compound that they might build to target the NMDA receptor, maybe it's going to help with pain management, maybe it's going to help with Alzheimer's, maybe it's going to help with depression, or maybe it'll help with these single gene GRIN disorders. And so, often the way that the drug development works is they want to, find something that they can prove to the FDA or other agencies that are trying to prove the efficacy of these drugs. And so they'll kind of pick the one that looks like it could work the best, and hope that there could be a bigger market after. So they're very interested in working with us. And I can't really get into the details now, but there's at least one test that's about to start with animal models that exist around these GRIN genes using one of these therapies.
I think that was the final episode of your podcast, right? That you have somebody has successfully created a mouse model. I wonder if you could tell people more about that, how that works and what it enables. Is the mouse is it effective or reminiscent in any way? I know in some neurodevelopmental disorders, it's uncanny how much similarity there is between the mouse traits and the human traits, but in others it couldn't be it couldn't be further from what it's actually like. What has your experience been?
Yeah, so I mean, there are probably about a dozen mouse models that have been made with different kinds of GRIN disorder that mirror the human condition. And so in the podcast, I was talking specifically about the one that was made with my son Bryson's condition. And so we got some preliminary results in January. And so that was sort of just before COVID shut everything down. So I would say in terms of like phenotyping the mouse behavior, they hadn't really gotten to that yet in terms of how similar it would be to Bryson and the other kids that have his precise variant. But there certainly are similarities just generally with the animal models and the kids with GRIN. So, the most obvious one is that the epilepsy is very common with these people who have GRIN disorder. and many of the mice models have epilepsy. They are certainly weaker. There's this one test that is done on loss of function mice here in Toronto where they hold them upside down and try and get them to hold onto cages and see how long they can kind of hold themselves by wrapping their paws around the wires on a cage. The mice that have the variants have a lot of trouble with that. So so there certainly are some similarities.
I'd like to actually cover this in more detail because I know you have written and talked a lot about the concept of a search for a cure and how this is actually not, it's not as straightforward as many people think. It's a really personal issue for many families, how they approach this. I think it'd be great to hear from you about how you think about it and also some of the progress you've made speaking to different researchers, companies about gene editing and other of these kind of drug repurposing type approaches that you mentioned earlier.
Yeah. So, I mean, to start with, in terms of I guess the ethics of cure, let's call it, it's not something that I'd really thought about before I started working on this podcast, started doing all of the research. And, I still remember the first time that I put something out on Twitter saying, like, I'm looking for a cure for my son, Bryson, and another parent that I know of a child with a different rare disease tweeted back to me and say, like, why a cure? Like, what is it about a cure? What does that mean? And so that really got me thinking a lot about this. And when I was putting the podcast together, we decided to do like a whole episode just talking about, the ethics around the idea of cure. And it is quite complicated. And it's something that I'm still kind of thinking about and working my way through a lot. And part of it is because, many people with disabilities don't like the term at all. They feel like it kind of minimizes their existence. So, if you talk about a cure for someone with cerebral palsy, it can be an offensive term to them. There are people who are in wheelchairs and, feel that it's ableist to think that they should want to walk if they can get around just fine in the wheelchair. They feel like it's the world that should be, changing, that we have so many barriers to accessibility that we should be working on taking those down instead of having this focus on trying to change them. Now, Bryson's situation is different because he's got severe developmental delays. He's not really able to make up those kinds of decisions himself about what kinds of treatments he wants. So as his parents, we kind of have to do that for him. So, some of the things where I've kind of landed on it is that it is the right thing for me to do. Like, it is a good thing for me to do as a dad, to try to do whatever I can to help Bryson, to give him more agency, to be able to express his wants and desires, to be able to get around by himself. Right now he's in a wheelchair and we have to push it. I would love if he can either walk or push his own wheelchair, tell us when he's in pain and why he's in pain. But at the same time, I feel like I do have to put in the work to try to change the world and not just him. And so I'm trying to learn a lot more about disability politics and get involved in trying to, improve the world, not just for Bryson, but for everyone with disabilities. And one example is just around accessible washrooms for adults, right? So, and obviously, many washrooms have wheelchair stalls. And I think there's been a big campaign actually in the UK where you are, to try to have more adult change tables and more washrooms. And I think the UK is a lot further ahead than where we are in North America in that regard. So there's a group that I'm just starting to get involved in that's really active in trying to promote that within Canada.
So go ahead, please.
I was just going to get to, the other part of the question. Remind me again, it was where we're at in our search for.
Exactly. Where you are in your search.
Yeah. So I would say that we're still pretty early. At CureGRIN, we've decided that with the money that we're raising so far, what we want to do is focus on what we're calling foundational research, which is, the basic things that are going to make everyone, whether they're doing research at universities, hospitals, or pharma or biotech companies, more interested in researching and studying GRIN. So we're investing in animal models. We're investing in research to try to identify biomarkers that could help recognize if a cure or therapy is working. And we're investing in patient registries and natural history studies. So that's kind of where we're focusing on right now. Our plan over the next year is to kind of build a consensus among families, researchers, and clinicians on what are the key questions that we need answered in order to develop cures and therapies for GRIN disorder. And so instead of the traditional model of a foundation where you kind of raise money and ask researchers to come to you with how they think they could spend your money in the best way, we want to prioritize how we think the money should be spent, and then either go out and find the right people or have an RFP on a very specific area to get people to come to us on how they're going to answer that question. This is something that there's a researcher called David Fajgenbaum of the Castleman Collective who's published around how this can work. And so we're kind of using that model in our attempts. So I think we're really still at the stage where we're laying the foundation, but certainly feel like we've got really good momentum. CureGRIN's only been around for a little over a year, and we feel like we've come a long way already.
Do you have a good example of the kind of requests for a proposal or the patient-led research question that you all have in mind or thinking of? Because I think this is a really powerful concept, rather than saying we have a couple 100,000 researchers, please write grants and proposals and let us know what you want to work on, and actually saying here's one or several of our priorities. If you think you can solve them or make progress, then write to us. I think that's really powerful. Do you have any examples already picked out or ideas?
Yeah, I would say we don't really have any picked out yet, but I can give you an example. And part of it is that I think that there's been many examples that we've heard from other rare disease foundations where what the researchers think a cure looks like is different from what families and patients think a cure looks like. And so a big part of it is, we're going to be doing this shortly, but going to all of our community and saying, like, what are the most important elements of helping your kids, of therapies and cures for your kids that we should be focusing on? So asking them, and for some, they're going to say that it's epilepsy. For some, that they're going to say that it's helping my kid to be able to talk or at least to be able to communicate what he wants. Some are going to say that it's around improving cortical visual impairment. So a lot of our kids, their eyes work perfectly fine, but the way that the vision gets processed in the brain is jumbled. So how can they get help with that? And so part of it is really identifying those kinds of things. But that, examples could be around those, but they could also just be around, we may say, okay, we know that you could have a gene therapy that can target the brain, that can repair the damage that the variant causes, but we don't know what part of the brain is the most impactful. Is it the hippocampus? Is it somewhere else? So, an example of the kind of research we may do could be around doing a bunch of different tests on small animals, or even potentially it would need to get to primates, which have a bigger brain, and figuring out where do we need to target to have the best effect.
You all have received some funding from the Chan-Zuckerberg Foundation, which is started by Priscilla Chan and Mark Zuckerberg, and they have an amazing amount of funding available specifically for rare disease, which I think is great. I wonder if you could tell us a little bit more about how you work with the foundation, how that process went. I think you were probably one of the first groups they awarded funding to, and I guess you're probably supplementing that with fundraising that you all are doing as a group as well.
That's right. Yeah. So we've raised, and our families have been great in terms of raising money for helping us find therapies and cures. But the Chan-Zuckerberg Initiative, they did just over a year ago. They did a request for proposals from rare disease foundations, and it's interesting, like the way that they're choosing to invest their money around rare diseases. A big part of it is not around funding actual research, but funding patient organizations and helping us to build capacity. So they initially had planned on funding 10 different organizations, but they got hundreds of applications and decided to fund 30. So we were one of the first 30. It's a 2-year program, and I think they're planning to fund more groups in the future. Everything's been changed a bit, because just as the program was about to start was when coronavirus hit. And so, there were going to be a lot of in-person conferences and that kind of thing. So it's really become a lot more virtual than the original plans, but it's just a fantastic thing, because it's funding, but it's also a lot of training and capacity building that just really is helping us get stronger as an organization. So, everything from how do we get better at just our governance and our finances, to how do we become more effective and make sure we're investing our dollars more wisely as we invest in a search for a cure. So, so the money that we're getting from Chan-Zuckerberg Initiative has some stipulations around it where, they don't want us to spend that actual money on the research. They want us to spend it on building capacity for our organization and building a global network of researchers and clinicians. And so, that's what we're doing. And it's been great so far.
I think it's really smart, because it's like planting 30 seeds, rather than hauling in the oak trees and saying, chop them up, and spend it on research and then we'll see what happens. But they're seeding a network of patients and, like you said, training and those sorts of things that mean you can create a sustainable foundation that can run for a very long time, rather than putting all of your hopes onto a small number of research projects that, as we know, most research hypotheses are wrong, and so there's a huge degree of trial and error. You mentioned COVID there, and actually someone asked about this in the chat about the mouse model, and I was interested in how much that has affected not just the research that you all been doing, but operations more generally. Have you been able to respond and do most things as a group remotely, or has it slowed things down as labs are shut and some priorities are shifted?
Yeah, it certainly has slowed things down. So a lot of work that needs to be done in actual labs has been cut completely. Some of our researchers have had to cull, euthanized cages of animal models because it just wasn't safe for them to have enough people in the labs.
Right.
And so, fortunately, every strain or every actual variant of animal model has been kept. So we haven't, lost too much, but all of that kind of research has been on hold. I know most rare disease organizations say that their donations and funding have really suffered. We're only in our second year, so it's hard to really compare. But I imagine that we're behind where we would have been otherwise. But in terms of the group functioning, we're all in different places. So the two co-founders, I'm in Toronto. Denise is in Denver. Jillian, who's a board member and really active, who was on Jillian Hastings Ward, was a guest on this podcast. That's before. Right. Yeah. So she's in the UK. So we've got board members in the Netherlands, and so we're all over the place. So we're kind of used to working virtually. So we've been able to kind of continue on with that. A lot of the meetings, we were planning to go to San Francisco and meet with researchers there. We were able to do most of those through Zoom. So it's not quite the same, and there's more challenges, but I would say for the most part, although the research is on hold, we're able to continue moving forward.
Right. I actually wanted to ask you a question related to Jillian, because I've had the pleasure to interview quite a few very inspirational parents like yourselves who've dedicated themselves to research. So Jillian being one of them, also Matt Might and Nick Serow. You also mentioned David Feigenbaum earlier. I was wondering whether you have mentors or people like that have been coaching you on this mission, because I imagine this is one of the most unique skill sets in the world, right? To build and grow one of these things from scratch. So you have to understand the science and the finance and how to deal with people all over the world working remotely and all kinds of things. Have you found other people like that or are able to help you go on the journey?
Yes. And I would hesitate to even mention one because I think there's just been so many. But I would say, for the most part, it's been, we've met them through organizations like Global Genes. We've met them through Rare as One. And there's just such a spirit of giving and collaboration. So, whenever I have a question and I say, who can help with this? Someone will say, oh, I know this person over here.
Right.
It's done this already. And that person is always willing and able to jump on a call to discuss it. So that's one of the greatest things about it is just how much this community wants to help each other. There's, fortunately we're all doing our own thing, but we're not competitors. We're all in this together.
That's great. I'd like to shift over to the Q&A now because we've had a couple come through. One of them I think is just amazing that she's found her way into the webinar. But Shilpa says, my daughter is diagnosed with GRIN 2A and we're from London, UK. How can we be part of the CureGRIN Foundation for any research work?
So that's great. Good to connect with you, Shilpa. To find us, we're at CureGRIN.org. We've also got a fairly active group on Facebook. We would love to get you involved. I don't know. So we have a parent advisory committee. We have a number of subgroups from that committee, including groups that focus on science and research, and would love to get you involved. So is it okay if I give my email address here too? She can reach out directly. I'm Keith@CureGRIN.org. Would love to hear from you or anyone else.
Great. We have two questions from none other than my mother, Denise Short. With sequencing becoming more prevalent, are they discovering more cases of Bryson's condition? It's a great question, Mom. I was actually thinking of asking that one. And then the second one is, do you think there are misdiagnosed people, as in people who may have the condition that have been told it was something else, who may not have received the appropriate testing?
Yes. So for the second one first, there's definitely a lot of people who were told initially that the doctors thought it was something else. In Bryson's case, no one really speculated, but I've heard many stories of other people who thought it would probably Rett's or different things that, and sometimes, these are things that could be more degenerative. And so that those false diagnoses that aren't based on actual genetic reporting can be scary and not very helpful. On the first question, so we're constantly seeing more families getting diagnosed with GRIN disorder in the different genes, and specifically with Bryson's variant. I think we were amazed when we first found another person with his exact variant, an adult woman. I think she's 30 now, who has Bryson's variant. And, through the podcast, we meet more and more. And I think in the podcast, maybe at the end, we're up to 6. And I think now we know of 9 families in the world who have the same variant as Bryson's. So that's great. And the more people there are with a single variant too, the easier it is to kind of identify the phenotype of the variant and compare that to the mouse model. And it's great to have that growing community.
I wonder if you can explain to people just how that works and how vast the array of different possible variants is. So I don't know how many amino acids the gene has, but it's a big string of amino acids. And my understanding is it's not that everybody has it's not that everybody has the exact same variant, as is the case in most rare diseases, but it's a missense variant in the case of this disease, which is doubly challenging because it's not that the gene is truncated and you lose the protein, but it's actually that the protein is just subtly changed. And every family's, for the most part, the majority of people have a completely different protein, but in a different way. Is that right?
Yeah, that's right. And so there's 2 categories of it. The first one is, that there are 7 different genes that encode the NMDA receptor, so 7 different proteins. the NMDA receptor is a tetramer, so four of those proteins coming together. So there's at least four of those genes that we know of, or four of those genes that have pathogenic disease-causing variants. Possibly more, but there's at least four that are confirmed. And then within that, so my son's variant is in the GRIN1 gene. There are, I think, I don't know. For all of the genes, there's roughly 1,000 amino acids, give or take. And within each gene, there could be variants in any amino acid. There's some amino acids where there could even be different variants. So, in Bryson's GRIN1 gene, where there should be, in one of the amino acids where there should be a glycine, there's an arginine. But it's also possible, in theory, that glycine could be a different amino acid that might have a different impact. So yeah, and all of these variants can have a different impact on how, the structure. So in Bryson's, a glycine is a tiny amino acid. I mean, obviously they're all tiny, but it's an extra tiny one.
Tiny by comparison.
Tiny by comparison, with almost no electrical charge, where an arginine is, relatively a very large amino acid with a positive electrical charge. And so one of the things we don't even know yet, for example, is when these come together in an NMDA receptor with two GLUN1 proteins, so the proteins made by the GRIN1 gene, if there's two of these, ones with the mutation, two with the variant, can they even coexist together, or are these two positive amino acids or positive arginines close together? Can they even coexist, or do they just blow up the NMDA receptor? So there's still so much that we don't know. And in fact, one of the things I was thinking about is, as we're going along with this, we've got maybe 1,000 patients in the world that we know of. Only a couple hundred have been written up in the literature, and we think we know a lot. But then you compare it with COVID-19 and what we're seeing now, where there's millions of people in the world, and tens of thousands, if not hundreds of thousands, that have been written up in scientific literature. And we're still finding out new things every day. Like, it's amazing how little we probably actually know about GRIN.
Yeah, and I think that's a big part of the challenge, right? Is that every time you peel back a layer of the onion, it's more complex, and you have to go back to the lab, which can sometimes take years. Are you excited about the prospect of gene editing? Because this has come on the scene relatively recently, and only very recently has it been considered a potential therapy route for rare diseases. I was curious of whether you all have commissioned or looked at any research around whether the condition is potentially reversible, or whether I know it's not degenerative, but whether you can restore some kind of function potentially through editing. Is that something you've looked into at all?
Yeah, it's definitely something that we're looking into. And the indications are that in theory it should work. One of the things that we talk about in the podcast is that using CRISPR or using gene editing on in the blood or in the eyes or the liver. These are areas where it's easy to kind of use gene editing and to kind of spread around and get to everything. Delivering it to the brain is much more difficult. So we feel like it's probably something that is many years away, somewhere between 10 and 50, if I can give an enormous range. I think that it's likely that sooner than that we'll see small molecule therapies that are more likely to help our kids. But that kind of gene therapy really is the ideal situation, because if we can get it to enough NMDA receptors, it could potentially, and especially if you do it with kids, it could potentially get to the point where it would almost be like they don't have this disease at all. Right. So it's something that, if you think of a cure as being something that is fully restorative, that takes the individual back to the wild type, this would be something that would do that.
Amazing. We had a really great question come through, another one from Jessie. So she said that her family, dad, uncle, and three others have been affected by a rare condition, potentially something called Loeys-Dietz. And she says that one of the challenges she faces if she goes to the doctor, they add something like scoliosis to the chart, goes to another doctor, they add something completely different to the chart. And over time, it becomes a complete mess. You have this spiraling database of symptoms that actually is unusable for the doctors and unusable probably for the for the person as well. And so she asked a pretty open-ended question of whether you have any ideas of how we can change this. I think Jesse's in Canada, but anywhere in the world, I know you have ideas around how patients can take back more control of their data and make sense of it as a group. Sorry, Jesse, he. I called Jesse she. Thank you for correcting me. I wonder if you have any thoughts on that.
Not necessarily. I mean, one of the things that it reminds me of, so we're at this point now where there's a number of different there's a number of different researchers that have started registries for GRIN disorder, and we're at the point where we're trying to bring them all together and kind of evolve them to a natural history study. And there's a handful of companies that do natural history studies by sort of getting access to the patient records, and they use natural language processing to kind of scan all the patient records and develop a natural history study based on it. The reason why I think of that is because I'm always curious, like if we were to actually do that with Bryson, would the results be anywhere similar to what my wife and I would report if we were to say it ourselves? I just don't know. So yeah, I can definitely see that being a problem. And I wonder if that could be the case even more where, the gene has sort of an unknown significance. If there's a gene that's identified that's contributing to a disease, but it's not the only gene. And so, it could relate to a number of different things, but I definitely see how that could be a concern.
Absolutely. So I'm just conscious of time here. We're coming close to the hour. If anyone has any last burning questions, they can definitely put them into the chat. To close out on my side, I was wondering if you could just give one piece of advice for parents who, maybe are in the position you were in 5-10 years ago, thinking about dedicating more time and maybe potentially a career shift into research. Is there anything that you'd recommend people think about if they're in that situation?
No, I mean, it's tough. And I feel like I was in a privileged place where I was able to do this. And, I was able to, because I'd already kind of done podcasting and done journalism, I was able to sort of do it in a way where I could also kind of support myself financially. So most people can't do that. It is a lot of work. And so, if you're able to, I definitely encourage you to do it. You can't do it alone. You got to find other parents that you can team together with. But the other thing I think that's important to say is that, taking care of our kids with rare diseases is so much work on its own. And if you don't have the time or energy to do this, that's okay. Like, it's so hard to be a caregiver of a rare disease patient anyway. And I think sometimes people have this guilt that they're not doing enough. Like if you're a caregiver of a rare disease child or parent, you're doing enough.
I think that's great. And I also remember from the podcast, you had some support locally from the government. And I think it's also important to note that this isn't available everywhere. And when we talk about, we spoke about disability rights and some of those things it seems to me like that's an essential piece of the puzzle as well, having federal funding to help with caregiving for children, parents, or whoever, because it's, I can only imagine how incredibly challenging it is to have a full-time job and be a full-time carer like that. So even I think what is it like in Canada at the moment? Are you entitled to a few hours a week or something like that?
Yeah, it actually depends by province, and in Ontario it even depends on your region. But Bryson gets 7 hours of support a week. We're not using any of it now because we don't want extra people coming into our home during COVID.
Of course.
But when things get back to normal, basically we had someone coming every morning helping him get ready to school. That was paid for by the government.
Great. Well, thanks so much, Keith. I really appreciate you being part of this, and also thanks to all of the live guests for your Q&A. This will go out on the podcast in a few weeks, but thanks to everyone. I think this was a really successful experiment. We're getting some thank yous through the chat for you, Keith. So thanks so much. I know your day's just starting, and I really appreciate it.
Thank you, Patrick, and thanks to everyone who joined and asked questions. Really appreciate it.